Hello. I forgot that I had my mic on and you could hear me whispering to Kelly, but I have to remember that I have my mic on and continue whispering to you because little man is upstairs asleep and I cannot wake him up. This is probably the second hardest class of the semester to deliver without my figures due to copyright infringement. So I could be wrong about that, but I'm intimidated by this. I've put it off until literally the last moment that I can. So here we go. And I don't know why, because this is actually one of my favorites. And I say that about a lot of things all the time, but I really love muscles, skeletal muscles in particular. The first time I fell in love with the human body way back in my first semester anatomy and physiology class was when we met the muscles, and it was because it was the first time like I could comprehend or visualize, imagine what these little contractile proteins looked like and the way that they were sliding past each other and like pulling this thing called the sarcomere together and contracting and shortening muscle fibers so that we can do all this super cool stuff. So that's the other reason I might forget that I have a mic on or not even care, is because I'm super passionate about muscles. I might just be screaming about it. Furthermore, like muscles, today my son and I moved like 6 tons of sand on our own because of muscles. Muscles are so great. All right, which type helped us move all that sand? Skeletal muscle. Why is it called skeletal muscle? Because it's attached to bone. So bones act as the levers that skeletal muscle attaches to, and it will pull on your bones to move your bones around and let you do various things. So together the two systems give you locomotion and movement. So skeletal muscle attaches to bone. That's what makes it skeletal muscle. Cardiac muscle is found in the wall of the heart. And we will talk more about this next semester. The last type that we have is smooth muscle, and this is in the walls of our hollow organs. And usually, and I know for sure, you've got a figure in your book that is comparing these 3 types and uses some keywords and shows you the histology. So usually we would look at a figure of that and we would write out all of the words. Today we're just going to write out all of the words. If you want to see what I'm talking about, the like striation. What is striation? It's this banding pattern of light and dark and light and dark and light and dark that you observe in skeletal and cardiac muscle. And the reason that we see that is because they're using the same microscopic unit of contraction, and it causes that banding pattern. We'll talk more about that in a little bit, but this is like a key word that we should know for skeletal muscle, is that it's striated. Skeletal muscle is the only thing that you control. So weird, like when I think about that, because I am such a control freak and I think I control everything. Like in my cerebral, I'm like, you control everything. From an like anatomy and physiology perspective, like the only thing that I have voluntary control over is my lower motor neurons and their effect on my skeletal muscle. So this is the only one that gets that keyword. Skeletal muscle is voluntary. Maybe that's why I love it, because I can control it. And some would argue it's the only thing I could control. I would argue that's not so, because you could be like having a panic attack and so your heart is racing, but like your cerebrum could override and you could realize you do not have to freak out. It's just a spider. Okay. And cerebrum could override. I mean, of course, it's like hypothalamus. It's then, telling your brainstem and like, so it works down the chain, but we're not getting into that discussion right now. Skeletal muscle, the only thing that's really voluntary. And what else? It's multinucleate. So that's another keyword you will often see. And what does that mean? It means that our muscle fibers have many nuclei, and that's because during development you get these little myoblasts that are individual cells with their striations, of course, and their individual nuclei. And during development they fuse together and become one long muscle fiber. And so that muscle fiber has many cells then. And this is another thing that I don't know. Like, okay, people, let's geek out for a minute. This muscle right here, strap guy, sartorius. And while the diameter of a muscle fiber is microscopic, one muscle fiber is as long as the muscle in which it resides. Look how long that muscle is. Holy moly, people, it's amazing. And that's the one that lets you sit crisscross applesauce. And crisscross applesauce is pretty good sometimes. So without sartorius, I would argue you couldn't do it. So yeah, that's another thing that we could say. What are the cells? They are muscle fibers, and they are really long. So the cells in skeletal muscle are muscle fibers. Okay, and they're really long, and they have many nuclei, and they're super cool. Okay, cardiac muscle is found in the wall of your heart, in particularly this layer called the myocardium. The cells are called myocardiocytes or cardiomyocytes. Myo, muscle, cardio, heart, site cells. So these are the contractile cells that we find in the wall of the heart, and these are striated like skeletal muscle was, but they don't have huge long fibers, and they might have 1 to 3 nuclei. But they are little like branching cells. And so for our other keywords, we could say that these are striated. They are 1 to 3 nuclei, so they can be single nucleate or they could be multinucleate. And they are myocardiocytes. They're branched cells. And let's just imagine that here in blue we have one myocardiocyte, cardiomyocyte. And here in green we have the next. It's going to be joined to its neighbor. Histologically you'll see some separation. In the body it's not so. But it'll be joined to its neighbor. This is the other one. By this specialized structure. Does anybody remember what it is from way back in chapter 3 when we were talking cell junctions? It's called an intercalated disc. And there are 2 parts. What are they? The physical junction, desmosome that holds them together, and the chemical junction, gap junction, it's the pore between the cells. And so right here, histologically, you'll see it's a little bit darker here. And I'm sure that the assistant will show you this. So you'll see this like light banding pattern, and it'll be pink or gray, depending on how the slide is stained. And there will be these little tiny lines that are slightly darker, and that's an intercalated disc. So that's a specialization about cardiac muscle, is that it's the only place in the body that has intercalated discs. And what that is a gap junction and a desmosome. So the desmosome gives us our physical connection, and the gap junction gives us our electrical connection. I'm not going to write that all out in the interest of getting through this as quickly as possible so that little man doesn't wake up. You can pause it and write it down, rewind, slow the whole lecture down to 50%. And then I'm like normal. I don't know what my problem is. I'm working on it. Okay, anyway, so here are striations. Beautiful. Anything else we need to say about this? I don't think so. Smooth muscle is smooth. We find it in the walls of our hollow organs, and it does not have these striations that we observe in our other muscle tissue types. And we only have like single cells with one little nucleus, and since they're not striated, they're smooth. That's why they're smooth muscle cells. And what could we say about them? These are also, oh, and I didn't say this about heart. Heart's involuntary, or cardiac muscle is involuntary. Smooth muscle down here, making me think about that. Smooth muscle is involuntary. It's smooth. The cells are single nucleate. And again, we find it in the walls of our hollow organs. So the lab assistant will take you through all of the histology. You can also look at that in your book, which will be wonderful for your learning, I'm sure. Okay, characteristics of all of our muscle tissues. So not all of our muscle tissues do the same thing, right? What does skeletal muscle do? Well, it pulls on the bones and causes movement. What does cardiac muscle do? Well, it contracts the heart and causes the movement of blood so that the blood that enters your blood vessels. Okay, what does smooth muscle do? Well, if it's a smooth muscle in your stomach, it squashes all your food and mixes it with gastric juice. So if it's a smooth muscle in your blood vessels, it will constrict or dilate depending on which organs need which we need to be served at the moment. So really, like, our muscles, they're all moving something. That's something that they share in common. We just saw some differences. This one's the only voluntary one. Really, they're gonna be a lot different when you get down to, like, not horribly different. Yeah, they're gonna be a lot different when you get down to the nitty-gritty functioning at the physiological level. And that's because they're doing different things. But they do have some characteristics that they all share. That was a really long tangent for what probably would normally take you 30 seconds in the class. It's okay, take a breath. What muscle is that? Muscle of the day number 1, respiratory diaphragm. Characteristics of muscle tissues. So this is what all of them share. And then, like, after this, we're not going to talk about cardiac and smooth muscle until next semester. The entire rest of this lecture and all of next class is going to be on just skeletal muscle tissue. So this is awesome, but it's also a lot of information coming to you right now in your living room at the end of the semester in just 2 lectures. So use that skeletal muscle, which is excitable, as is cardiac muscle and smooth muscle. All of our muscle tissues are excitable, which means that they can be excited or aroused to a stimulus. So the only way that you can do that is if you're responsive to some kind of stimulus, say like a neurotransmitter coming from a lower motor neuron, something like that. Yeah, definitely like that. Excitability is the ability to respond to a stimulus. I bought all new markers, and that little man was supposed to put all the rocket markers right back there. Not give them to me anymore. My apologies, people. Excitability, the ability to respond to a stimulus or to be aroused to a stimulus. Okay? So cardiac muscle's cool, it sets its own pace, and you don't need the nervous system for your heart to run. It's pretty awesome. Now, if you want to be able to adjust your heart rate, given something like a bear running in your door, would a bear run in my door? We're not even going to go there right now. It wouldn't. But if a bear was in the yard and I were to freak out, if the bear and the burglar were together, I would probably have a sympathetic nervous system response, and then my nervous system is going to kick in and override what's happening naturally in the heart. So it's able to be excited by bears and burglars alike. And skeletal muscle is excited. It's aroused to a stimulus, or it's by a stimulus to contract. So smooth muscle, excitable. So that's a characteristic that all of our muscle tissues share. They can be aroused by a stimulus. They're able to respond to stuff. Like dead keratinocytes on the top of your skin can respond to nothing. They're dead. I mean, you could say, well, falling off is a response. Okay, if I scratch it off. Fair enough? Fair enough. Contractility. This is the ability to shorten and generate tension. So contractility is the ability to shorten or contract. Ability to contract. There are some contractions in which you do not shorten. You stay the same length. What are you talking about, lady? I'm talking about right now. If I were to push against this wall with all my might, I could generate as much tension. Watch. Oh, I'm not moving. Oh, but I'm pushing harder and harder from 1 foot all the way up to 1 forearm. And my muscles aren't changing length. So shorten isn't necessarily the best answer for that, because when you're pushing the wall, there's no change in the length of your muscle. You know why? I'm not moving that wall as hard as I might try. So the ability to contract, we could say, or generate tension. to contract. We'll just leave it at that. They are all contractile, which is the ability to contract and generate tension. Don't worry, we'll talk about the different types of muscle contraction later. Probably at the end of next lecture. Okay, so all of our muscle cells can, and shortening is the general rule. Okay, so yeah, when your smooth muscle cells, that's not quite shortening, but they all like bundle around and all of the cells get squeezed, and I mean, it is like shortening. Muscle, cardiac muscle also shortening. Skeletal muscle most of the time shortening, but you can also generate tension while your muscles are contracting, I guess is the point. They all display the ability to contract and generate tension. That tension is going to move stuff. They also actually all display the ability to be extended or extensibility. So contractility, we could think of this as being able to shorten so that you're less than resting length. Okay, and extend, the general rule. Extensibility, this means that you can actually lengthen and go beyond resting length. So all of our muscle tissue types can do that. So these can stretch beyond resting length. Okay, well, like we kind of all know that. I have been tighten, like moving all this sand today. All my muscles are tight, so they're just being squeezed down to lower than resting length. But I could stretch them out to resting length, and then I could stretch them beyond resting length a little bit. They'd feel great. Cardiac muscle also. If you have an increase in venous return to your heart, you have to be able to accommodate that. And so if you're using more of your blood, muscles, it'll squeeze on your veins and it'll push more blood back to your heart, and your cardiac muscle will be able to extend a little bit so that we can have this extra accommodation. And we all know that there are plenty of time when we eat far too much, and then our smooth muscle in our stomach can extend to accommodate all that, right? So extensibility is the ability to stretch beyond resting length, and that's another characteristic that they all have in common. Next is elasticity. This is the ability to bounce back to resting length. Elasticity, right? We've already talked about elasticity when it came to elastic protein fibers in our connective tissues, and we said that's what was special, is that it's the ability to stretch, sure, but then bounce back. All of our muscle tissues are going to be able to stretch or be extended, they're so short and bounce back, but then also when we contract, they're going to be able to bounce back to their resting length. So all of our muscle tissues display elasticity, or the ability to bounce back to resting length. Let's continue and talk about our muscle functions. Okay. So when we think about our muscle functions, the functions are going to be different depending on which muscle tissue type you're looking at. The functions of all of them, one function that they all share is that they help with movement. Okay, what does the skeletal muscle move? Perhaps your skeleton. So when skeletal muscle contracts, it pulls on bones. Wonderful. What does cardiac muscle move? It moves blood, right, through your cardiovascular system. So cardiac muscle is responsible for the movement of blood, and smooth muscle is responsible for movement as well of whatever it is passing through that organ. So blood vessels are organs. So blood is in your blood vessels. So the smooth muscle and cardiac muscle share that in common, is that they can move blood, sure, if the organ that we're talking about is blood vessels. So whatever's in the organ. So smooth muscle will move blood in blood vessels, but smooth muscle will move food and gastric juice in your stomach. It'll move all of that chyme and our various digestive enzymes, brushboard enzymes, and accessory secretions. All of that will be moved through the small intestine by the smooth muscle in that organ. So smooth muscle is going to move stuff, and what it's moving is dependent on which organ you're looking at. So that's really the only function that they have in common, is moving stuff. And you're like, yeah, lady, but they're not even moving the same stuff. That's fine. They all have movement as their first word. Okay, as far as other stuff, we'll talk about cardiac muscle and smooth muscle next semester. So what we're really going to focus on today is skeletal muscle. Not only today, next class as well. And so as far as other functions of skeletal muscle go, I mean, these two, they're wonderfully, fantastically boring, but also wonderfully fantastic. I dare you to live without them. I don't. You can't. This one is wonderful because it does so much awesome stuff. Other than moving your bones, it helps you to maintain posture. So that, like right now, I'm glad that I can like see my shadow so that I don't forget that I'm slouching. Like I can be my own like posture corrector, not even charge myself, because I can flex flex some of my muscles, my flexors. I can contract all of my skeletal muscles and maintain my posture. So I can do this voluntarily and all of a sudden think about it, or I could be falling asleep in class on my couch. But if we were in a real class, this would be more serious. The consequences of you falling asleep in the front row of class in a real class, huge. The consequences of you falling asleep in the front row of this class, you gotta start push play again and try and remember, where did I fall asleep? Do I have to listen to her talk about sphincters again? I don't wanna. So okay, maybe I didn't fall asleep there. Maybe you just blocked that part out, who knows? The point is that you could be falling asleep in class, either format, and the only way anybody knows you're not falling asleep is because your skeletal muscles maintain your posture. I am not lying to you when I tell you that I made my entire endocrinology semester in the front row falling asleep every day. Taking notes while falling asleep every day. No, not every day. Tuesday and Thursday at 8 a.m. endocrinology, and in the front row because I'm, that lady. And so here I am, like, keeping my eyes open, and all of those notes, like, because you don't want the instructor to know that you're falling asleep, so you keep your eyes open. And I would start a sentence and it was legible, and then the notes would just fiddle off. It was like this. Skeletal muscle. So I was sleeping all during that part, but my professor never knew it because my skeletal muscles were maintaining my posture with the best of them. So yeah, I mean, and what's unfortunate about that is that endocrinology is one of the classes that I did not get an A in. I'm not going to tell you what I got, because anything less than an A is an F to me. But the point is, afterwards, that was the class that made me a teacher. So you know who's an A in endocrinology now? All right, so that's something that is a very important function of skeletal muscle, helping you maintain your posture. Cardiac smooth muscle, they don't really do that. Okay, another thing that skeletal muscle does that the other two joints do is help you to stabilize your joints. So if you recall, last class we talked about joints and your synovial joints, which are awesome and reinforced by reinforcing ligaments and things. But if we look in here, everything that we see is not a muscle. Attached to muscle, sure, but you can have some muscles, and the muscles that, like, they're helping. Was this the best example of a joint I could use? Yeah, because it was on my table. But you will find that outside of your synovial joints, you'll find your skeletal muscles attaching and helping to stabilize the joint. So that's pretty cool. That's the other function of skeletal muscle. Oh my goodness, stabilizing joints. There we go. Now this is where we're gonna spend most of our time. because yeah, sure, cardiac muscle gets its own chapter next semester, but does it even get as much time as this one? Yeah, it does. Two lectures. So we'll have 2 lectures on skeletal muscle. Next semester I'll see you for 2 lectures of cardiac muscle. Smooth muscle doesn't get an explicit lecture because we find smooth muscle in the organs that we're going to talk about in the other systems that we'll talk about next semester. So we'll get there in just a minute. Okay. As far as our skeletal and muscle anatomy goes, the skeletal muscles are all served by nerves that contain the lower motor neurons that are going to branch to form motor units that help us control our muscles for varying tasks. And then, of course, it's a living organ, so we have to have some arteries and veins to give us some blood supply. So, some little man just woke up. Let me make sure everything's good there. We'll come back to lecture. Blood supply goes, every muscle obviously has to be supplied by a nerve and blood vessels. It's living, so it needs an artery and a vein. Most of our named arteries, muscular arteries, are going to branch into the specific arteries that serve various skeletal muscles. So be aware that our skeletal muscles are served by arteries and veins, which bring them their continuous supply of oxygen and nutrients. The nerve is going to contain, we'll say, lower motor neurons that serve various motor units. So our nerve contains lower motor neurons serving various motor units. And we'll talk more about what a motor unit is in just a little bit. Connective tissue sheaths. This is like a real nitty-gritty anatomy thing. That just as our nerves were surrounded in various connective tissue sheaths, so too are our skeletal muscles. So if you look at a skeletal muscle, like all of these models have them imaged as red, and it's because they're red, but then they die and they look brown. But they're very dark in color. They have very, very highly vascularized. So in order to be able to contract and do things like that, you have to have good nutrient blood supply. You got a lot of blood vessels. So another thing that you have are all of these various connective tissue sheaths. And if we're thinking about the entire muscle, then the sheath that is surrounding it, just as we had epineurum on top or upon a nerve, we have epimysium is upon a muscle, muscle. So my epimysium is on top of the muscle, and it is going to contain all of the fascicles of muscle fibers that make up that muscle. So, and it's going to be able to resist tension in many directions. So what tissue type might that be? This is dense, irregular connective tissue. Yeah, bub. Okay, well, let's work on your skeletal muscles, and you'll be able to figure out how to button it. So left hand is going to grab one hand, or grab one half of the button. Right hand is going to grab the shirt. Left hand. Well, that's like your whole body popping out. Come down here, sit down, and try and button those buttons while I finish lecture. You gotta get it done before family dinner. So just come sit here. Watch. All the muscles are going to work together. You're going to use the muscles in left hand. Or is it right hand first on boys? It's right hand's going to be first. Okay, you're going to take all the muscles in right hand and grab the right side of one button. And left hand is going to hold the fabric in the hole. And right hand is going to stick the button through. And they're going to work together and they're going to get the job done. Okay, within our skeletal muscles, we have groups. You can do it, or if not, I'll do it in a minute. Or guess who just came out and can help you. Groups of muscle fibers are gathered together, just like we had fascicles of neurons, we have fascicles of muscle fibers, and they are surrounded in perineurium. This is muscle. Perimysium. Perimysium. And perimysium is also going to be able to resist tension in many directions. So perimysium is also dense, irregular connective tissue. My love, I love you. I can do it if you really need me to. But I also have to teach class. Within these fascicles of muscle fibers, we have muscle fibers. And muscle fibers have voltage-gated channels. They're conductive. So in order to keep this muscle fiber electrically insulated from this muscle fiber in the fascicle, from this muscle fiber in the fascicle, each of my individual muscle fibers has to be surrounded in its own connective tissue sheath. Yes, dear.
I'm looking for the bunny suits.
Okay. They're not in there. So then this innermost connective sheath, just as in nerves was the endoneurium, this is the endomysium. And what type of tissue was it in the neurons, or in our nerves, insulating our neurons? It was areolar connective tissue, right? And so, yeah, that's what it is here. Insulating individual muscle fibers, we have endomysium, which is areolar connective tissue, and that's its function. Can you just get him some money for snacks? They're somewhere in one of those. I'm almost done. I feel like it. Maybe not. Nice. So, epimysium, upon the muscle, dense irregular connective tissue, resists tension in many directions. Perimysium surrounds individual fascicles of muscle fibers, also resists stretch in many directions. Individual muscle fibers are electrically insulated from their neighbors in fascicles by endomysium. No, they're in a box, or actually in the front pocket of the lunchbox right there on the bar. And this is areolar connective tissue. And its job is to electrically insulate our muscle fibers. Okay. Wunderbar. Sissy's got them. Attachments, direct and indirect attachments. Direct attachments, I don't know, like you directly attach to a tendon, or you indirectly attach to tissues through subcutaneous. We're not gonna, I never worry horribly about attachments other than the specific attachments we need to know. Tendons attach muscles to bones. Muscles are attached to tendons by bones. Here's our bone. Okay? And here is a direct attachment to that bone, a tendon. Oh, and then what's cool, what type of tissue is that? That's dense regular connective tissue, right? Between the tendon and the epimysium, we have this stuff called fascia. And that's where you'll start to see this transition from dense regular to dense irregular connective tissue. Okay, so that's direct. You've got other types of direct attachments where, aponeurosis, where you have this, like, we're attaching one belly of the epicranial muscle, the frontal belly, across this wide place, wide span to the occipital belly. Same muscle, epicranius, upon the cranium, frontal belly, occipital belly, attached by the epicranial aponeurosis. That's an attachment. We're going to let you read the rest about it in your book. It's Sunday dinner time. I got this little guy here who's really hungry, eating his fruit snack. I got Sissy on the stairs. We have to get through, oh my goodness. We're not going to get through it before dinner.
We are.
Okay, we're getting through C2 sarcolemma. This is the plasma membrane of our muscle fibers, and it's specialized because it's got voltage-gated potassium and voltage-gated potassium. Voltage-gated potassium. It's got voltage-gated sodium and voltage-gated potassium channels down its entire length. That's what allows it to be conductive. So here, let's just pretend this is a muscle fiber. And so it's got voltage gated sodium channels and voltage gated potassium channels down its length. So here's our voltage gated potassium channel, voltage gated potassium channel. Okay, cool. Sarcolemma. That's what allows muscle cells to be conductive. All right, wonderful. Sarcoplasm. This is the cytoplasm of the muscle fiber. So everything that we find inside here, it's mitochondria, it's specialized organelles that are going to allow it to contract and be different than other cells. Everything is there in the sarcoplasm. Okay, so normally all of our intracellular fluid, all of our cytoplasm is low in calcium. In a resting muscle cell, it's also low in calcium. This is going to be important. The sarcoplasm floods with calcium when we're contracting. We're not doing that yet, we're relaxing. Believe it or not, this is relaxed. So sarcoplasm, this is the cytoplasm of our muscle cells, and it contains all of our organelles. And what are we talking about next? Modified organelles. Ooh, it was like I knew that was coming. Modified organelles, what are they? They are these super cool things called myofibrils that are super cool because they have myofilaments that contract. This is when I fell in love with the myofilament. And so we'll be there after Sunday dinner. I cannot talk right now. Myofibrils are going to contain our myofilaments. This is where I said I needed to get through. So here we've got our thick and thin myofilaments. These are going to be the structures that slide past each other to allow for shortening of a muscle, contraction, and generation of tension. Sarcoplasmic reticulum. So myofibrils, you're like, well lady, you didn't even draw it up here on the picture. Myofibrils are kind of hard to draw here because there's a sarcomere and we're going to get our banding and our whatever. Sarcoplasmic reticulum is modified smooth endoplasmic reticulum that is going to contain high intracellular levels of calcium. And I drew this extra line up here right now because over here we've got more sarcolemma continuing away, and this thing is this specialized structure called the T-tubule. So coming down here, here's a T, transverse. It's transverse to the axis of the muscle fiber that we're talking about right now. So this is a transverse tubule or a T-tubule, and what it is an inward extension of the sarcolemma that also contains our voltage-gated channels. And what this is going to do is it's going to allow for this action potential to be conducted deep into this muscle fiber. Okay, so our T-tubules, let's come there and just write down a definition. This is an inward extension of the sarcolemma. And now, the reason that I went ahead and told you that before I talked about sarcoplasmic reticulum is because we find sarcoplasmic reticulum, this modified smooth endoplasmic reticulum, arranged in this really specific way, abutting our T-tubules here. And that's going to be really important when we get into the microanatomy that leads to the physiology of a muscle cell. So sarcoplasmic reticulum, I'm drawing right here. And this is SR, or sarcoplasmic reticulum. Why is it sarcoplasmic? Because it's in the sarcoplasm. Why is it reticulum? Because it's smooth endoplasmic reticulum, and what its big function is to store high levels of intracellular calcium, or store increased calcium intracellularly. And if you recall, when we got to the transmissive segment of a neuron, it was the calcium entering that neuron and moving those regulatory proteins that allowed for exocytosis of our neurotransmitter. It's the action potential coming down here and causing the release of calcium from the sarcoplasmic reticulum, and then the interaction of this calcium with these myofibrils that is going to allow for muscle contraction to occur. So take a breath. Using which muscle of the day? Number 1, your respiratory diaphragm, which flattens during inspiration. And I will see you soon after Sunday dinner. Okay, people, it's bedtime almost. Little man's playing downstairs, so I'm going to try and go through this quickly review what I didn't talk about earlier. So we were talking about skeletal muscle anatomy, and that anatomy is going to get down to the microscopic level, and the microanatomy is going to lead us to come up with this thing called the sliding filament model of contraction. And the reason that it's a model of contraction is because you can't actually observe it happening in a living organism. And the reason that that is the case is because living organisms are alive, and this is microscopic, and it's like so teeny tiny that you can't actually observe it happening like in a muscle. Or last I heard you couldn't. What we can instead see is what a muscle looks like before and after muscle contraction. There's a figure in your book that shows you the difference, and we can see that, yeah, in a contracted muscle you shorten, and these things called sarcomeres, and you're going to have these filaments then we can say that are sliding past each other that are going to form the molecular basis for contracting a muscle. So I came back to this microanatomy slide to kind of review everything that's important as it relates to this picture that I have kind of pre-drawn up already so that we can be comfortable with it and know what it is we're looking at. I'm just gonna... So I drew this out a little more neatly than we saw earlier. This is the sarcolemma here in black. And so that's the plasma membrane of our muscle fiber. I only drew the voltage gated channels on this one segment here, but we have voltage gated sodium and voltage gated potassium channels down the length of the muscle fiber. At one spot on the muscle fiber where the lower motor neuron is coming into contact with that muscle fiber, we actually also have something called the motor end plate. So at one spot, the motor end plate, we've got neurotransmitter receptors here. We'll talk more about that in a little bit. Right now what we're trying to do is wrap our head around our muscle fiber microanatomy and how it's going to lead us to this notion that we can slide these thick and thin myofilaments I told you a little bit about past each other. Okay, so floating around inside this muscle cell is cytoplasm, because all cells have cytoplasm, right? Muscle, sarcoplasm, cytoplasm. Little man, are you running around? Okay. Inside then, this cell, it's got all the stuff that cells have. Particularly interesting is that these cells fuse, so we have many nuclei hanging out in here. They are highly aerobically active, so we've got many mitochondria hanging out in here. But what's unique about these cells that can contract is they've got some specialized modified organelles. A modification of the sarcolemma is these things that we already talked about, T-tubules, transverse tubules that are extensions of the sarcolemma that come deep down into the muscle fiber and surround these myofibrils. Okay? T-tubules contain voltage-gated sodium and voltage-gated potassium channels, so they're going to conduct our action potential deep into the muscle fiber. I wish I had a cameraman. Okay, all right, so sarcoplasmic reticulum, that's this stuff right here, SR, sarcoplasmic reticulum, and it's modified smooth endoplasmic reticulum. Again, this is one muscle fiber, so I've just drawn 3 different parts because it's hard to teach a class in your living room without figures. So we've got our myofibrils are all linked up under here, and like everything you see here is like here. So those are my myofibrils there with sarcomeres. Believe me, we'll get there in a minute. Sarcoplasmic reticulum is that modified smooth endoplasmic reticulum that allows the muscle fiber to store high levels of calcium intracellularly. So, okay, why does that matter? Well, because we're going to conduct this action potential down the sarcolemma and down our T-tubules, and when it comes down our T-tubules, it's going to open these voltage-gated calcium channels that are going to allow this calcium out. to flood the sarcoplasm. And it's going to flood the sarcoplasm and come and bind with these proteins in here that are forming the unit of contraction for our muscle and move the regulatory interaction so that what we can do is now slide these filaments past each other, shorten this, shorten every one of those bad boys along the entire length of the muscle fiber, shorten the muscle fiber, and contract ultimately a whole muscle is what you're trying to get to. Okay, so myofibrils are these little awesome modified organelles that contain our contractile proteins. I think on our last board I had written out thick and thin filaments. That's what I've drawn out here, and we're going to talk a lot about those. Now, myofibrils as their own individual modified organelle are so interesting and unique, they get their entire own series of slides. So let's get there. Myofibril features, this is what gives us striations. It's the banding of these light and dark bands that gives us this striated appearance. So this would be a dark band, and then there'd be a light band, and here'd be a dark band, and then there'd be a light band, and here'd be a dark band, light band, and so on and so forth. Down the entire length of the muscle fiber, we would see that. And that is coming because of the arrangement of these things called sarcomeres. A sarcomere is the microscopic unit of contraction. It's the smallest unit capable of contraction, and it's super exciting to me. I love it with all my heart and soul. So we're going to draw sarcomeres over and over. I've actually drawn one right here. This is a sarcomere. A sarcomere has a couple definitions. One I just told you is that it's the smallest unit of contraction. But another is that it goes from Z disc to Z disc. So I'm going to erase the rest of this and draw us up another sarcomere and talk about it. So if I ever draw something in a lot of detail, you should probably be able to recognize it and understand what everything in that figure is doing, because it's probably pretty important. Sarcomeres, smallest unit of contraction. Is one definition. Another definition is that they go from Z disc to Z disc. So we need to know some more anatomy. Here's the Z disc right here. Where? What the bleep are you talking about, lady? That is always the question. Almost always the question. This thing right here is a Z disc. And here's another one. Oh, it's too close. But whatever, we're going with it. Suspend your disbelief. It's howl time, people. It's 1 minute till 8. Pause. Let's see if I can get him to howl on camera that you can hear. Okay, well, it's time. Can you practice real quick on the way outside? It's time. Oh, oh, oh. Okay, let's go outside, but can you practice on the way up? Okay, so a sarcomere then is the smallest unit of contraction. It's the span from Z disc to Z disc. So here's one drawn out here. In a sarcomere, we find these overlapping filaments. I'm gonna burp. They're thick filaments and thin filaments. Cut. Not really. Okay. So we have overlapping of these myofilaments. These are the proteins that we find that are going to be sliding past each other in our sliding filament model of contraction. So I told you that earlier that myofibrils had myofilaments, thin filaments, and thick filaments. Now let's dive into those in a little more detail. These ones on this figure over here that are pointing outwards are my thick filaments. So these are all thick, and these contain the contractile protein myosin. So myosin filaments contain myosin, and these are our thick filaments. and they really only have myosin as the contractile protein. Okay, and that's it, nothing else. Well, okay, that's not necessarily true. If we look at it, this thing in the middle that I've drawn is what's anatomically called the M line. And what it is these proteins that are running perpendicular to the longitudinal axis of the muscle fiber and anchoring our thick filaments. So all of our thick filaments, our myosin proteins, are pointing away from the M line in 360 degrees around. And our myosin filaments, these myosin proteins, if you look at one myosin protein, it's like they wrap around and it's got this double-headed golf club kind of structure. So each of these thick filaments has just thousands of these myosin proteins in here, all pointing out these little myosin heads, and they're all pointing out and stretching away. And how I even got here was trying to also talk about these striations in this thick and thin pattern and of like banding that we see. And we're going to see that as we draw out the features of this myofibril. So I'm such a turkey. It's because I'm tired, my belly's full, rest and digest is working, not really my like cognition. Because I said thick filaments, the only protein is myosin. It's the only contractile protein, that's for darn sure. But there's another protein here that's super cool called titin. I think some books call it elastin. I know your book's calling it titin. And what it is this other protein that's not involved in contraction. This is what gives our skeletal muscle its elasticity, or striated muscle its elasticity. It's super cool. So that what happens when our muscle is contracting, we're shortening this sarcomere, we're pulling these Z discs together, the thick filaments are reaching up and grabbing onto our thin filaments and pulling it in, so that what you do is you compress this other protein. So what made me a turkey is I said that myosin was the only protein there. It's not. There's another protein. It's titin. And this compresses like a spring, so that when you shorten this sarcomere, we just like spring it together, and now there's a ton of potential energy. So as soon as we relax, as soon as we stop sending this signal from our lower motor neuron and we pump all of that calcium back into the sarcoplasm, then we can bounce back to resting length without requiring any energy. So titin is the protein, we'll say this gives the elasticity, or the ability to bounce back. Okay, so that's our thick filament. Myosin is the contractile protein, and we've got these myosin proteins. Like if this is my M line, then, and let's say this is neatly wound around each other like the figure in your book shows, there's one myosin protein. And here, pointing out the other way toward the other Z disc, is another myosin protein. Okay, well that's just like one. This is a thick filament. They're everywhere. And this filament is really thick. Okay? And so we're anchored to the M line by those kind of like tails, the tails. So there's my tail, and my tail is going to be like facing into the M line and anchored to the M line. So if we wanted to write a function, we could, for the M line here, we could say it anchors my thick filaments, and this is our center of our sarcomere. Okay, so here my actin filaments have three proteins that are really kind of important. You'll see in your book actin's like this globular thing, and it comes together to become like this filamentous. And so, and on each of these little actin proteins that we find in our thin filaments, we have these myosin head binding sites. Oh, what might want to grab onto there? Haha, myosin, right? So, okay, great. When our muscle is relaxed, my myosin head binding sites are covered up and blocked by this regulatory protein. It's actually like two proteins, but it's like a troponin tropomyosin complex. This like big long green thing right here would be my equivalent to, we'll call it tropomyosin. Tropomyosin. And hanging off of tropomyosin all over the place, we have these other little proteins called troponin. That has a calcium binding site. Okay, so attached to tropomyosin, I have these little troponin proteins. So troponin, the troponin tropomyosin complex is a regulatory complex that is blocking the myosin head binding site, or the active site, on actin. So in a relaxed muscle, there's no calcium out and around in the sarcoplasm. So troponin and tropomyosin are in this shape that covers up the myosin head binding site on that actin. And then when calcium binds to troponin, it shifts it and changes the shape so that now we can expose the myosin head binding sites on our actin molecules. So this is a question that I like to ask often, like true or false? The only protein in a thin filament is actin. And what's the answer? False. Because troponin and tropomyosin are proteins that we find in thin filaments. Now what if I said true or false? The only contractile protein we find in thin filaments is actin. Well, that's true. Okay, so be aware of that. All right, so then if we were to like zoom in here, let's say, and say this is my actin filament, my thin filament here, it's anchored to a Z disc. Okay, and they're facing in toward the M line. Okay, so there's a little spot in here that's free of actin in a relaxed sarcomere. That's called the H zone. Why do we care about these things? Well, because if you're looking at a slide and you have to identify is this contracted or relaxed, if you see an H zone, a spot that's free of actin, you're relaxed. Because when we start contracting, my thick filaments are going to be pulling out and they're going to be reaching and grabbing the thin filaments attached to this Z disc from here and the thin filament attached to this Z disc from here, and we're pulling them together. So at some point, my thin filaments are going to overlap my thick filaments and there's not going to be any spot that's free of actin. Okay, so in a relaxed sarcomere, that spot's free of actin, and then we've got this region here where I have overlapping of my thick and thin filaments, and that's the A band that looks dark. So when in our striations here, this was a long, really roundabout way kind of to get back to striations, but your A band is your dark band, and your I band is your light band. And how you can remember which is that the word dark has an A in it. So that's my A band. And what my A band is here is this is the spot where I have overlap of my thick and thin filaments. The length of my thick filament does not change. What changes is the length of my sarcomere, the distance from Z disc to Z disc. So when my thick filament is working, my myosin heads are reaching up and grabbing, and they're pulling on to these thin filaments and pulling it closer together, so my Z discs get closer together. So that means my A band is never going to change in shape. I tell you these details because I also used to teach the MCAT, and if you're going to take an MCAT, you're going to need to know details like this. So the A band is the band where we are dark. Our dark band is the A band, and this is where we have overlap of our thick and thin filaments. Okay? Now, in one sarcomere, you only see half of an I band. If we look in here, this white space then would be the I band, or the light band. What's great about the word light is that it has the letter I in it, and that's the I band. And the I band here's half an I band. In a relaxed sarcomere, now we have an area that's free of myosin. So if we were to look to see an entire I band, we would have to zoom in here to our next sarcomere, And then the half of the eye band here and the half of the eye band here would give us the entire eye band. And in that area we are free of myosin. Okay? And so that's never going to completely disappear because you're going to compress this spring-like titan here on the end. Okay? What else? M-line, H-zone, I-band, A-band, Z-disc, Z-disc. I believe that is all of the important anatomical structures we need to worry about. Molecular composition of myofilaments. I already went over that. So that's what we were talking about here. All the proteins we find. Awesome. So we're good on that part. Wunderbar. Sarcoplasmic reticulum and T-tubules. We've already talked about that as well. But what I didn't quite really fully explain was a triad. So, okay, what's cool is that you'll have sarcoplasmic reticulum does what? Stores high levels of intracellular calcium, right? So if our thin filament has this regulatory protein that has a calcium binding site on it, as soon as calcium enters the sarcoplasm and binds that, we're going to move that and we're going to start contracting. So we want to keep calcium high inside the cell, but we only want to let it out when we are contracting. So that's why we keep it high in the sarcoplasmic reticulum. And it's arranged around the T-tubules in this really kind of cool way. So if this is our sarcolemma here in black, and our T-tubules coming down here, we've got our voltage-gated sodium and voltage-gated potassium channels conducting the action potential down the sarcolemma. It's coming down the T-tubules and deep into the muscle fiber to surround my myofibrils. Okay, so that's really cool. Watch a video on this. They're great. And it's something really cool to see the way that they draw it out for you. I can't make you a video right now from my living room. She says that she's making you a video right now from in her living room. Okay, so anyway, I digress. Right next to each of these T-tubules, we have an enlarged region of the sarcoplasmic reticulum called the terminal cistern. So two are terminal cisternae. So terminal cisternae. And these are enlarged regions of the sarcoplasmic reticulum where we've got those voltage-gated calcium channels. And what's going to happen is this action potential is coming down this T-tubule. It's going to open up this channel, which is just a protein, and we're going to allow calcium now to come out and flood the sarcoplasm. Well, what's great about that then is that it can bind to troponin, move the tropomyosin complex. Now our active site on actin is free. Myosin can reach up and grab it, and we can start shortening our sarcomere. Okay? So sarcoplasmic and reticulum and T-tubules are found in these arrangements called triads. And what's happening here at that triad, this is a triad, we've got two terminal cisterns, cisternae, and one T-tubule makes a triad. So I've got one terminal cistern here, plus one T-tubule, plus one terminal cistern here. All of this is a triad, and what's cool is that as this action potential is coming down this sarcolemma, when it gets down this triad, I'm going to have this like easy access to a good, readily available supply store of intracellular calcium. Okay. Sliding filament model of contraction. This is where we will pick up next time, going in nitty-gritty detail. So let me just have you think about it this way, because it can be hard to wrap your mind around. So, and what you see in your body is a whole muscle contracting. So in this muscle, we have all these fascicles of individual muscle fibers. And this is what we're talking about, is a cell as long as this muscle. And when I contract right now, all these muscle fibers in these motor units and muscle fascicles are going to contract and shorten so that I can flex my forearm, okay? So this action potential is coming down a lower motor neuron and being conducted both directions away from the motor end plate. We'll get there next time. So it's going to spread like so quick down, and then I'm going to start this cascade of events inside the cell to lead to my thick and thin filaments sliding past each other, the sliding filament model of contraction. And again, we call it a model of contraction because we can't actually observe it happening in a living subject, like actually, we can just see like the before and after effect. So this is what it looks like before you have slid our filaments past each other, and then afterward, our Z-discs would be a lot closer together. So maybe our Z-discs would instead of being this far apart, after I slide my filaments past each other, I bring them in and I shorten here. Well, if I shorten every sarcomere along the entire length of my muscle fiber, I'm going to do a lot of shortening and I'm going to generate tension. So when I'm doing that, what's sliding is these thin filaments past these thick filaments. So let's just come back to, our thin filaments are sliding past the thick filaments. And how are they doing that? Here are these thick filaments pointing out this way. Here's this thin filament, which is only drawn as a line over here, but remember, it's this globular thing with myosin head binding sites. And so it just wants to reach up and grab it, okay? And there are lots of these around. So when I move the troponin-tropomyosin complex away, what happens is my myosin heads reach up and they grab it, and then they're going to pivot and pull it toward the center of the sarcomere. So it goes inward. And here was my Z-disc. That was probably too far away. So let's say here was my Z-disc before. Now I pull it and I just move in. So I hope that made sense. Watch a video. This is where we're going tomorrow. And so, yeah, that's it. Myosin is going to reach up and grab the myosin head binding site and slide my thin filaments toward the center of the sarcomere. So the thing is, we said a sarcomere was from Z-disc to Z-disc. A Z-disc I did not really define for you. Those are the proteins that are anchoring my thin filaments. And my thin filaments point toward the M line. So let's just imagine my fingers are thin filaments and they're thick, imaginary thick filaments in between here. What's happening, and my thumbs, let's say, are the Z-discs. As the thick filaments are reaching up and grabbing the myosin head binding site, they're pulling it, and they're pulling it, and they're pulling it, okay? So it's like pulling, okay? So as that's happening in 360 degrees around these myofibrils, and everybody's doing it, then I'm pulling my thin filaments toward each other, and then I'll have overlap, and I shorten my sarcomere, I pull my Z-discs together, and now I've contracted, okay? And when I had to teach MCAT, they said that I needed to come up with an analogy to help people understand this, and the one that my teacher used was something about a horse, and if you were trying to pull a horse, and I'm just like, it's not a good one. But I was watching Walking Dead at the time, and they happened to have a zombie in their well. And so the thing about this is if it's just one thick myofilament grabbing onto one myosin head binding site on one thin filament and pulling toward the center of the sarcomere and letting go, well then it would just bounce back. But it's not. They're all of my myosin heads are in different phases of what's called my cross-bridge cycle, and they're all working together to pull these thin filaments toward each other. So how does that relate to zombies and wells? Well, if you've got a zombie in a well, and you're able to get a rope down in there and hooked on around it, then if you were just to be alone and use both your hands and grab onto the zombie and pull and let go, yeah, he'd come up, but then he'd splash back down and he'd probably break to pieces, and then you've really contaminated your water. So instead of pulling and letting go and pulling and letting go, what you do is you get your other zombie hunter friends behind you with their two hands, and you cross one over the other, and everybody's in some different phase of pulling that rope to get that zombie out of that well. That's what's happening with my thin filaments. My thick filaments are reaching up. The myosin head binding sites are reaching up. Sorry, the myosin heads are reaching up and grabbing onto that myosin head binding site and pulling it toward the center of the sarcomere and it's happening all over the place. Seriously, watch a video about it because it's so cool. This is literally when I fell in love with the body, because I could just see it like, oh my goodness, these molecular components, teeny tiny molecular components of this cell lead to my ability to do this, and to do this, and to do this, and to do this, and say, read it over in your book, review it. We'll come back for next class to go over this in nitty-gritty detail. You look pretty bad back here, Kelly. Here, let's put it together. Let's hold it together, okay? Okay, man. Hold it together. Literally, okay. Great. Stay put. Yes. Don't say no to me, Kelly. Okay. It's got the same proteins, they're just arranged in a different way. Like macrame around the pot. I hate macrame, but we just got macrame in my house. So let me actually use this as a perfect example of a real life demonstration. So in our cardiac and skeletal muscle, we have these contractile proteins that like band in this like striating pattern like that. And in smooth muscle, it's the same proteins, but they're not banding in like a linear way. They're like squashing it around like this. So that imagine like if this was a cell, not an alloplant, if instead this was a cell and I could squash it, when they contract, when these cells, when these contractile proteins are working, they are squishing this from all the different angles. I now understand exactly why we have been reorganizing and cleaning and everything this whole weekend, so that I could give you that real life example right in that moment from my most awesome living room, office, coronavirus classroom of the year. Okay, I digress.